Hi, welcome to Biology Classes with Lakshmi Chandra. Today we are focusing our attention on yet another topic of genetic disorders. It is color blindness. Color blindness. The term itself tells you what the disease is. Color blindness means a person is not able to distinguish between colors. Not all colors, but some colors. So in order to understand the basis of that, why some people are not able to distinguish between colors, we should know what is making our eyes to distinguish the colors. What helps our eyes to distinguish the colors. So to distinguish the colors, our eyes have got a sort of cells called as cone cells. It is termed as cone cells. So cone cells are present in our eyes and its property is to enable color vision. If cone cells are not functioning in one's body, then we'll see everything as black and white only. That means cone cells are actually enabling us to distinguish the colors. But these cone cells are of three types in the human eye. There are three types. You call it as S-type, M-type and L-type. What is this S, M and L? All these distinguishes are made with respect to the wavelength of light. You know the primary colors red, blue and green, they have got different wavelength range. Red is having the longest wavelength and so the cone cells which can actually perceive this longest wavelength or they are sensitive to the longest wavelength, you call such cone cells as L-type. So here L refers to long waves. So cone cells L-types, what does it do? They are sensitive to red light. And I have written here the word opsin. Opsin is a pigment present for color vision. Cone cells have got a pigment responsible for color vision and those pigments are called as opsins. Okay. So these opsins are of different types according to the wavelength of light that it can perceive. So if the cone cells are of L types means it is perceiving what kind of a wavelength? Long wavelength. So that means it is aiding in the vision of red light, red, orange, such wavelengths. Now you can say what is this M? M is medium type, medium type of wavelength can be perceived using M-type cone cells and that means it is sensitive for green light. And yet another type of cone cell is called as S-type, that is very short wavelength. Short wavelength it can actually perceive, it is sensitive to and so it is able to perceive blue light. So cone cells are of three types depending upon the nature of wavelength which it can perceive. So accordingly it is called as short type or S-type, M-type or medium type and L-type or long wavelength type. So long wavelength for red, medium for green and short for blue light. So that's it. Now we'll come to the genetics part of this one. This was the basics that I've told you. Color blindness is caused due to the malfunctioning of these cone cells. If in our body these cone cells S, M or L type they are not functioning properly or if they are absent, the person will become colorblind. Okay, now we'll focus on to the genetic reason behind this. So in order to understand the genetic reason behind this, we should go behind the genes responsible for this and where are those genes present, in which chromosome they are present likewise. So here also, as I have stated here, the L-type and M-type cone cells, the genes for these cone cells are present on the X chromosome. And the gene for the S-type are present, they're present in chromosome number 7. So if a person's body is not having M-type or L-type cone cells, that means the person it is something responsible, something with respect to the X chromosome. So if it is with respect to the X chromosome, it becomes sex-linked disease. Because I have told you in the previous videos that X and Y chromosomes are the sex chromosome. So if it is responsible, if X chromosome is responsible, that means what kind of disease this will come under? Sex-linked disease. In the previous videos, we had learned other sex-linked diseases as well, the genetic basis of that, the inheritance pattern of that, everything we had covered in the previous videos like thalassemia, hemophilia, sickle cell anemia, all these things we had already covered. So if you have not gone through, please subscribe the channel so that you can just go through one after the other and you have a clear basis of what these diseases are. Coming back, here X chromosome is responsible for this. So this disease is what? Sex-linked disease. Now, this is also termed here, another point of color blindness is that it is sex-linked. Another one, it is recessive disorder. This was also explained in the previous videos. What is the meaning of recessive disorder and how it is differing from dominant disorder? See, recessive disorder means a disorder which will happen in one's body only if both the chromosomes responsible are defective. Both the chromosomes. So in the case of in human body, you know, males are XY and females are XX. Okay, so this defect is with respect to which chromosome? This defect is with respect to the X chromosome. This defect is with respect to the X chromosome. Which defect? M and L type cone cells, if they are absent or if they are malfunctioning, it is all because in this chromosome that particular gene is not present or that particular gene is not functioning properly. That means it is linked to X chromosome. That means it is sex-linked disease. So X chromosome. In the case of males, if this X chromosome is defective, the male is surely to get the disease. But in the case of females, if say one X chromosome is defective, you have the other X chromosome which is normal, which will have the normal gene, which will not allow this particular individual to become colorblind. Because only one X chromosome is getting affected, the other one is normal. So when will this female become colorblind? Only if both the X chromosomes gets affected. Only if when the genes in both the X chromosomes are absent, this particular female will become colorblind. That means for this a female to become colorblind, both the X chromosomes should be defective. That means it is a recessive disorder. If one X chromosome is defective and the other one is normal, the normal one will dominate this one. So the individual will not get the disease. Okay, so what kind of disease is this? Recessive. Recessive means it will affect the individual only if both the chromosomes are defective. What about males? Males, there is no question at all because it has only one X chromosome. If that is defective, the male will get the disease. So when you take the ratio, the number of colorblind males are more than the number of colorblind females. What is the reason? Simple, because it is a sex-linked and it is a recessive disorder. Sex-linked means which one? X-linked and recessive disorder. Okay, since it is affecting the X chromosome, males are having only one X chromosome, whereas females are having a pair of X chromosomes. There is always a chance that males, if it is defective, they will get the disease, but females, both the X chromosomes should be defective, only then they will get the disease. So the number of colorblind males are always more in number than the number of colorblind females. So I hope these 2 points are clear to you. Now when a person's body is not having M cell or L type cone cells, what will happen? They cannot sense it, they are not able to visualize which one is red color or which one is green color. They cannot distinguish between red and green because that wavelength is not actually received. It is received but it is not perceived. It is not sensed by the cone cells because that cell is not at all present. So the person will have failure to discriminate, to identify, to distinguish between what? Red and blue color. This is 90% of the colorblind people have this particular defect. What defect? They are not able to discriminate between red and red and green color, not blue color, green color. This is not blue color, green color. Please change this to green color, not blue but green. Red and green color. Red and green because M and L type of cone cells are not present and both these are there near to each other in the X chromosome. But then, and this here I've written here Daltonism. I hope you all know about the scientist Dalton. Okay, Dalton, the scientist was also colorblind. He couldn't distinguish between red and green color. So that is because cone cells M type and L type are either absent or defective in his body. Okay. Now come to the other type, children. See, S type. In this S type, if this S type cone cells are absent, or if the S type cone cells are defective in the patient's body, in the person's body, then it is due to the defect in chromosome number 7. Because the gene responsible for S type cone cell is present in which chromosome? It is present in chromosome number 7. It is present in chromosome number 7. Okay. So in this case, males and females are equal. They have equal chance of getting this particular disease, and this is not sex linked, but this is recessive. Okay, so chromosome number 7, this is not a sex chromosome. It's not at all a sex chromosome. And so what will happen? Since this is not a sex chromosome, both males and females have equal chance of getting the disease. They have equal chance of getting the disease, and in this case, if it is M or L type, males are having a chance of getting the disease more than that of the females. I hope till here it is clear to you. Okay. Now, for normal vision, all these three cells should function, isn't it? Short wavelength, medium and long wavelength. All these three cells should function for normal color vision. So if all the three are functional, then you call it as trichromacy. Trichromacy. Trichromacy means all the three cells are functioning properly in the organism's body. So you call, if all the three cells are functional, you call it as trichromacy. Trichromacy. If all the three are not functional, cone cells, all these three types are not functional at all, then you call it as monochromacy. Monochromacy. Then how will the vision be? Black and white. The person can see a thing as black or white only. No color vision is possible because all the cone cells are absent. So that is monochromacy. So if all the three are functional, it is trichromacy. And if all the three are absent, what it is? It is monochromacy. So two new terms, chromacy, trichromacy, both three are functional. Monochromacy, all the three are absent. They are seeing it as black or white. That's all. Okay, this is the condition. It's a condition when all the three are present and all the three cone cells are absent. This is the condition. Now let us look at this particular tabular column, here. You have mentioned here cone cells, L type, M type and S type. If L type cone cells are missing, L type is missing, L type cone cell is not at all present, then the disease is called as Protanomia. Protanomia. Protanomia is a disease affecting our vision. In what condition? When L type of cone cells are missing. L type is missing, the disease is Protanomia. L type is present. Imagine L type is present but it is not able to function properly. That means L type cone cells are defective. L type are defective, then it is called as Protanomaly. Protanomaly. Some abnormality. So that is it. Protanomaly. Defective. It is present but defective. Okay, now the next case you see M type. M type cells are missing. You call it as Deuteranopia. Deuteranopia. What is Deuteranopia? M type cells are absent. Okay. Next one, if M type cells are defective, it is called as Deuteranomaly. So anomaly, this is anomaly, this coming. That means some defect. Okay. Protanomaly, L type is defective. Deuteranomaly, M type is defective. And if S type is defective, what will you call it as? Tritanomaly. Tritanomaly. If M type is missing, you call it as Tritanopia. So you see here, Tritanopia. Clear to you this one? So cone cells are 3 types. If they are missing, the disease is Protanomia, Deuteranopia or Tritanopia. If it is defective, it ends with Mally. Protanomaly, Deuteranomaly or Tritanomaly. It's likewise. I hope this is clear. See how I have written here absent and Achromatopsia. Achromatopsia. What is this absent and Achromatopsia? All the 3 cells are missing. The disease is called as Achromatopsia. If all the 3 are non-functional, it is called as Monochromacy. If all these 3 are absent, it is called as Achromatopsia. I hope it is clear the difference between these two. Monochromacy means cone cells. All these 3 cone cells are not at all present in the body. It is absent. Oh, sorry, it is not functional. Then you call it as monochromacy. If it is absent, not at all present, it is absent, then you call it as Achromatopsia. Achromatopsia. So these are new terms related to color blindness, which you should be very clear with. It is very easy to understand. If there is any defect, it ends with Mally. Protanomaly, Deuteronomy likewise. Otherwise, it ends with Mia or Pia likewise. Okay, so missing means it is likewise. So you can understand it very clearly. So this is it. So regarding color blindness, it is all related with our cone cells. 3 types of cone cells. Their absence, presence and what the disease is. Now we will move on to another part of this particular color blindness, that is genetic transmission from male to female and all genetic transmission we will move on to. Okay, so let us see how it is getting passed on from parents to progeny. Let us take 2-3 conditions and accordingly we will understand. So in the first condition, father is affected. First condition I am taking, father is affected. So males, so I am putting here X, Y. Okay, father is affected. So I am putting here XC. XC, colorblind father. Okay, colorblind father crossed with normal female. XX, female is normal. Then what will be the condition of the progeny? Whether the boys will be colorblind or the girls will be colorblind, likewise we will see. So here gametes. So X, C and Y. This is one set. Another set of gametes will be X and X. This is Punnett square. Regarding the construction of Punnett square and all, it was all dealt with in the previous videos. How to draw the Punnett square, what is Punnett square and all. So in the basics of genetics which I have taken, it is all mentioned very clearly. If you have not gone through, you subscribe the channel, go to the playlist and you will get one after the other. It is written topic wise, it is written. It will be easy for you to fetch up and just go through that. The Punnett square. Okay, so here XX chromosomes and XCY. So I have split up this gametes, male gametes here and the female gametes here. Okay, now I am filling up the box XCX, top and left. Here also top, Y and X. Here XCX, here XY. XY or YX, anyway you can write. So you can have a look here. XC is coming here. See, females. But what about males here? XY, they are normal. XY here, male is also normal. Because in the case of males, always males will have a Y chromosome coming from father. Males will always have Y chromosome. Look here, Y chromosome coming from father. And X chromosome is coming from the mother. So X chromosome, both the X chromosomes which is coming from mother is normal. So males will be normal. So in this case, when the father is defective, the male child will be always normal. Now in the case of female child, one X chromosome is coming from father and another from mother. So the one which is coming from father is always always defective. Okay, so in that case, one is coming from father, another one is coming from mother. So one chromosome is defective but the other one is normal. So this particular child, this female child, will the child be colorblind or will the child be normal? Yes, the child will be normal. Why the child will be normal? Because even though one X chromosome is defective, there is another X chromosome which is compensating for the loss. Okay, the other chromosome is normal. So it will produce normal cone cells, and that normal cone cells will have all the three types, S, M and L type in them, and it is sensitive to all the three types of wavelengths. So the female child also here, the female child also will be normal. At the same time, the female child is a carrier of the disease. The female child, though it is normal, the female child will be carrier for the disease. It can transmit the disease to the future generation. Okay, why this female is not defective? Because this is recessive disorder. It will become defective only if both the X chromosomes are defective. So in the first case, when the father is colorblind and the mother is normal, the children born will all be normal, but female child, they will be carriers. They will be carriers of the disease. This is the first condition. Now let us look at the next condition. Let's look at the opposite case. The males are normal and female is colorblind. For the female to be colorblind, both the X chromosomes should have the disease, isn't it? Only then the female will be colorblind. If only one X chromosome is having, then the female is what? The female is carrier only. So our second condition is male is normal, his father is normal, but mother is colorblind. Okay, opposite. So in this case also I am drawing the Punnett square. XY father, then XC the female gametes, here. Okay, so in this case progeny will be XXC, XCY and XCY. I hope you understood how I have written this. See, just in each column you fill up the left and the top gamete. That's it. Left XC and X, likewise you fill up. So now what is the result here? This is female. Is the female colorblind? No. Female is normal. Isn't it? Female is normal. Here also female is normal. But what about male child? Male child is colorblind. Why male child is colorblind? Because male child is getting only the Y chromosome from the father. The X chromosome is coming from the mother. You see, mother is colorblind. If any one X chromosome the child is getting, the child is surely to be colorblind. So in this case, this males are colorblind, both cases, but females are normal or carrier. So our conclusion is males will be colorblind, but females will be normal or carrier. See, to come to this particular conclusion, you need not go on writing this particular Punnett square and all. If it is a 1 mark question, if this is asked, how easily you can write this? Suppose the question is, father is defective and mother is normal. Father is defective and mother is normal. What will be the condition of the male child? So imagine male child is getting only Y chromosome from the father. So Y chromosome is normal for the father. In the question itself it is written what? Mother is normal. So male child will be normal. What about female child? Since mother is normal, one X chromosome that the female is getting from the mother will be normal only. So female will also be normal. Likewise, you can just apply a little bit logic and you can write this answer very easily. Here I am drawing this and showing to you so you can understand it better. Okay, so this is the condition. So 2 conditions we have completed. First condition was we have taken father is defective, and the second condition we have taken mother is defective. So in both cases we found that female child is coming normal. In both conditions we found female child is coming normal. Now then in which condition that female child will get the disease? Imagine father is colorblind. Father is colorblind, XCY, and mother is carrier. Mother is normal, but she is carrying the disease. One X chromosome is XC, I have written. In that case, how will be the combination? XCY. Male gametes have split up. I am going to split up the female gametes. XCX I have written. Female gametes also got splitted up. Okay, this way it got splitted up. Now filling up the boxes. XC, XC. You look here, both X chromosomes are defective. So here the female child is colorblind. But in this case, XC, female child is going to be normal. Here, so in this case, the female child is affected and here the female child is normal but carrier. Here, it is XCY and XY. So here see, the male child can be defective or diseased and the male child can be normal. So in this case, there is every chance that the female child is getting affected and the male child is also getting affected. At the same time, there is also a chance that the female child is normal, male child is also normal. So this particular combination, when father is diseased and mother is a carrier, there is every possibility of getting a defective child or a normal child, whether it is male or female. Okay, now let us look at the last possibility, possibility number 4, where both the parents, that is male as a father as well as mother is getting affected. Father as well as mother is getting affected. So XCY and mother is also getting affected. Okay, so both X chromosomes in the mother is having the defects. So both X chromosome is denoted by XC and the father XCY. In that case, what will be the probable progenies, whether they will be diseased or not, whether female or male? That case, let us just examine here. See, it is XCXC. So female and female is defective. Here also XCXC. Again female is defective. In this case XCY. Male is having the disease. Here also XCY, male and male child is having the disease. So in this case, when the father is diseased and mother is also diseased, the child born to them will have color blindness, whether it is male or female. Okay, so we have completed the 4 different combinations, 4 different possibilities of getting color blindness. Getting color blindness means of genetic transmission from parents to progeny. So it's very easy for you to write. And so let us conclude. So color blindness is a sex-linked recessive disorder caused due to the defect in chromosome number in X chromosome, But except in the case of blue color vision, that is in the case of chromosome number 7 alone, this is all affecting our cone cells, and we have learned the different diseases associated with it and also the genetic basis of transmission. Okay, so with today's topic, I have taken almost all the topics in our NCERT textbook related to genetic disorders. That is, we have completed sickle cell anemia, thalassemia, hemophilia, phenylketonuria. Okay, and today we completed color blindness also. So in the next session onwards, we'll deal with chromosomal disorders, the reasons behind that, whether the different names given to the chromosomal disorders, okay, and how is there any possibility of getting cured, or how we can just overcome that, what is the genetic basis of that. All these things we'll be dealing with in the next video. Thank you for now.